Good evening. Now we will discuss in continuation to our gene cloning and expression. We will discuss a strategy for the production of recombinant protein in E. coli. So first question is, why do we need the protein? So as you all know that proteins are used as a therapeutics like a growth hormone, like hormones, insulin, vaccines, etc. Used as enzyme for various purposes like restriction enzyme, polymerase, protease, lipase, all these enzymes are required. For research also we need good amount of protein with high purity to characterize that protein. As you know that genome sequencing is easy nowadays, but characterization of the genome or the genes present in the entire genome is difficult. For that also we apply this, means we need recombinant protein so that we can characterize it. Now what are the sources? How can we get desired amount of protein with good quality of protein? So either we can purify from the endogenous sources, or we can apply recombinant DNA technology, or we can call it molecular cloning. So serious limitations of protein produced from endogenous sources are quantities. We will not get always sufficient amount of the protein because nature gives a limited amount of protein. Most of the proteins are in limited amount in their native host. Purity is also a problem. When we purify, that protein is not remain isolated. They are associated with their other partner protein. So whenever we purify, it's difficult to purify your particular protein alone. Other protein may also comes along with your protein. And consistency, because in growth condition of organism in different environments are different, so consistency also matters. So it will not be consistent, means purification or isolation methods, quantity, all these will vary. Suppose we are isolating some protein from the plant, so if plant is growing in Gujarat or growing in Bihar, their content will be different. So consistency will differ, and isolation methods will differ, quantity of that protein will differ. Despite of same species, if they are growing in different environments, so composition will differ. So that is a problem. So we apply recombinant DNA technology method. If we are applying recombinant DNA technology method, so we use particular host and we grow that host in a controlled environment. So we can get more amount of protein because we are particularly cloning and producing that protein in our desired host. So we add a strong promoter, as we have already discussed in expression vector. We have a strong promoter, so production of that protein will be more. Purity, so since we add our particular tag in that protein, affinity tag, so purification will be also easier in comparison to the native sources, native host. And consistency, since we are growing it in a controlled environment, controlled, you can say medium, known temperature, so almost amount and you can say other things will be consistent, constant. It will not change much. These are some of the examples of therapeutic protein, recombinant protein, which has changed the all, you can say, biopharmaceutical landscape has been changed. You can see now we have this data of since 1982 to 1999, but now you can see this is the old slide. So you can now imagine hundreds of therapeutic proteins, recombinant proteins are in the market. So we have already discussed about this, how can we clone, in previous lecture. So we can isolate our desired gene. How can we isolate? We have discussed in that in a strategy of gene cloning. So no need to again repeat this. No need to repeat this chapter also. What are the players of gene cloning? Why we should use E. coli? We have already discussed. Now, okay. So as you know that uh, we have to apply this step, as we have already discussed that just let me change this place if it will change. Okay, so what we do, we isolate our positive clone from the E. coli host, cloning host like DH5 alpha, DH10 beta, and then we transfer again that recombinant vector positive clone into the E. coli expression host. As we have already discussed about expression host, so mainly we use BL21, that is first generation, you can say, expression host, in which lone and OMP protease is deficient. So we isolate recombinant plasmid from the colony harboring positive clone, and then we transfer it into again competent E. coli expression host. And then after transformation, as you know, it will grow into a colony next day. So we take 1 to 4 colonies in different test tube with media and antibiotics, and then we allow it to grow overnight. And then we again second morning, again we take 4 tubes with fresh media, and then we again inoculate from the overnight grown culture again. So that is called secondary culture. When this culture, secondary culture, will grow up to 0.6 to 0.8 OD, then we add IPTG, 0.1 to 1 millimolar IPTG, which is an inducer. So before adding IPTG, we must take 1 ml from each tube, so that we can differentiate uninduced versus induced. Again, after induction, means after adding IPTG, we add we allow it to grow for 3 to 4 hours or sometimes overnight, and then we take, you can see here, we can take 500 microliter to 1 ml from each uninduced versus induced all, and then we lyse that cell. We first harvest that cell using centrifugation, and then we add directly 2x SDS buffer, and then we boil it, so that total cell will be brushed and all protein will be in that. So that is called cell lysate. Now we take all this cell lysate and then we run the protein through SDS page. So from this data, so after SDS page, we stain it with Kumasi brilliant blue dye, so we can see this band. So from this SDS page, you can say from this gel, you have to determine whether you got induction or not. So you can easily see here, this is IPTG, minus means in which we have not added IPTG, that sample, and plus means in which we have added IPTG. So you can easily see that in plus sample, IPTG added sample, there is one band which is thicker, you can say intense band. That means ki our gene induced inside E. coli host, new host. Once we confirm that that induction is proper, our gene is getting expressed, we can again perform same thing using the colony. We have taken here 1 to 4 colonies, so that sometimes some colony may not show induction. So if we get induction in all colonies, so no need, we can prepare a glycerol stock and keep it, and then depending upon our requirement, we can again start from the single colony, and then we grow it in a desired volume, 1 liter, 2 liter or whatever. That is a normal steps of protein production. We can produce recombinant protein in E. coli. Now this is not easy, there are multiple hurdles in production of recombinant protein in E. coli. As you know, E. coli doesn't allow post-translational modifications. So if your protein required disulfide bond formation, if your protein requires glycosylation, phosphorylation, so E. coli will not allow that. And major problem in E. coli host is inclusion body formation, means your protein may be misfolded and form a particle like structure, misfolded aggregated structure, that is called inclusion body. So that inclusion body, since it is considered as a misfolded protein, so it is not useful. Although you are producing more amount of protein, but that protein is not retaining its normal conformation, three-dimensional structure, so it will not show activity. So two major problems: no post-translational modification and inclusion body formation. Sometimes we don't get expression at all also, less expression we get. So now, since this is a 5-decade journey of recombinant protein production, scientists have solved most of the problem, but still some problems are existing. So under five subheadings, we will discuss the strategy to produce more amount of recombinant protein and good quality of recombinant protein. So we will discuss by changing host strain, how can we produce more amount of recombinant protein, good quality of recombinant protein, by changing the vector, by changing culture parameters, by co-expression, and by altering the gene sequence. One by one, we will quickly go through it. So now, most of the scientists, they prefer this PET vector, since it has very strong promoter, T7 promoter, and it has very strong affinity tag, that is his tag. So when you clone this gene of interest, your gene of interest in this vector, PET vector, so since PET vector has T7 promoter, that promoter is not recognized by any of the RNA polymerase of the E. coli. Means promoter is strong, since it is viral promoter is strong, but RNA polymerase should recognize it as a promoter, then it will start transcription. So if you are using BL21 strain for production of recombinant protein and your gene is cloned in the PET28 vector, you will not get protein, because any of the E. coli RNA polymerase will not recognize. So there is no transcription, no translation, no protein production. So what is the solution? So scientists have added T7 RNA polymerase coding gene in the E. coli BL21. As you know, BL21 is a basic strain in which lone protease and OMP protease gene are mutated. So this is a protease deficient strain. Since that protease deficient strain, T7 RNA polymerase coding gene has been added, means integrated in the genome, and that gene is called DE3 gene. So now this strain is called BL21D3 strain. Means you have to use BL21D3 strain if your gene is cloned in the PET vector, PET28 vector having T7 promoter. Now, so this strain is very good strain, but sometimes people observe that leaky expression. So leaky expression means what? Leaky expression means without addition of IPTG also there is little expression. Means when we are not adding inducer, then also there is expression. That is called leaky expression. So leaky expression is considered limitations, because sometimes your protein may be toxic to the E. coli. So due to that toxicity, E. coli will not grow. So everything is fine, but if E. coli will not grow due to toxicity, so your media is not utilized optimally. Means cell number is not increasing, so production of protein will be minimized, since number of cells are not growing. And you know that growth of E. coli doesn't mean the size of E. coli is increasing, means number of cells are increasing. So due to toxicity, if your protein is toxic to the E. coli, then you will not get sufficient cell number in a particular volume, unit volume of your media. So this is loss. So to overcome this, scientists first of all they try to find out the region. So you can see here in absence of IPTG, in absence of inducer, there is little expression of this T7 RNA polymerase gene. So it binds to the T7 promoter here, and so they start producing this target protein. Target protein may be toxic, so due to toxicity this cell will not grow. So this is called limitations. So to overcome this, next strain designed was T7 this BL21D3 P-lyase strain. Means lysozyme coding gene has been cloned in a plasmid, and that plasmid has been added in this BL21D3 strain. So this T7 lysozyme is inhibitor to the T7 RNA polymerase. So if there is a basal amount of T7 RNA polymerase present in the cell, that will be inhibited by T7 lysozyme. So there is no availability of free T7 RNA polymerase, so it will not bind to T7 promoter, so there is no production of toxic protein. So now you can say that if there is no production of protein, so our all purpose will get vanished, because our all purpose of this cloning is to produce recombinant protein. So what happens when we add the IPTG, when number of cells are sufficient, when OD reaches up to 0.6 to 0.8 OD, then number of cells are sufficient, then we add IPTG. So what will happen? This IPTG binds, and then they start rapid production of T7 RNA polymerase. But lysozyme gene is not expressing that much. That is their production is constant. So now suppose there are 5 lysozyme protein, 5 molecule per unit time. So they are capable to bind or inhibit only 5 molecules of T7 RNA polymerase. So 95 is free. Now they are capable to bind. So there is rapid production of your protein. Since number of cells are sufficient in the media, now even that cell is not growing, number of cells are sufficient, so production is sufficient. So if we are facing the problem of toxicity, then we should not use BL21D3, we should use BL21D3 P lysozyme. Even in lysozyme, there are lysozyme E, lysozyme S. I'm not going to discuss in that detail now. So now sometimes or most of the time when we produce recombinant protein of eukaryotic origin in E. coli, so there is codon biasness. So due to codon biasness, there is no production of or less production of your recombinant protein, or sometimes truncated protein production due to codon biasness. Now, what is codon biasness? So you have seen, you know this effect. What is the effect of codon biasness on production of recombinant protein? But now, what is codon biasness? So from chart you can see different organisms have different preferences of you can say synonymous codon. As you can see in this chart, arginine is coded by 6 codons. So in ideal condition, all codons should be preferred equally by different organism, all organism. But you can see AGA is in 22% of arginine coding sequence in human, but in Drosophila 10%, in E. coli 1%. Means AGA codon of arginine is not preferred by E. coli, but it is preferred by human. Same way, CGU codon is preferred by E. coli, but not preferred by human. Means there is codon. This discrepancy, this difference in preferences of the codon sets, synonymous codon set, is called codon biasness. So due to codon biasness, what happens? Due to codon biasness, when you clone the human gene, so there is chances of this AGA codon is more in that gene. But since E. coli doesn't prefer that, their genome doesn't have that much amount of AGA codon for arginine, so tRNA which decode this AGA codon, their number is also less. That tRNA, you can say it rare tRNA, and this codon is called rare codon because they are not preferring, E. coli is not preferring. So since E. coli doesn't prefer this AGA codon, so during translation, tRNA having this anticodon of this AGA will not come quickly to the ribosome. So what will happen? Due to scarcity of this tRNA, rate of translation will get slow, or sometimes due to much delayed translation may get aborted, so you will get truncated protein. So now what is the solution? This is a problem when we produce human protein in E. coli, so that E. coli may face this effect of codon biasness, your protein production is less. So scientists have prepared a E. coli strain in which they have added extra gene which codes for this rare tRNA. So there is no scarcity of rare tRNA. So now your translation will go smoothly. So this initially Strategene company has produced two strains, in which you can see here one strain is called BL21 codon plus real strain. So this E. coli cells contains extra copies of the RGU, isoleucine Y and leucine W tRNA genes, which recognize the AGA, AGG, AUA, CUA codon, overcome the problems of AT reach gene of eukaryote. Second strain they produced BL21 codon plus RP strain. Competent cells contain extra copies of the tRNA gene arginine U, which recognizes the AGA, AGG arginine codon, and the tRNA gene proline, which recognize the proline codon strain, overcome the bias of GC reach. Means depending upon whether your gene is of AC reach genome or AT reach genome or GC reach genome, accordingly we can choose the E. coli strain for production of recombinant protein, because this E. coli strains has extra copy of that tRNA, rare tRNA. So we have to choose. So next company, Novagene, they produced the E. coli strain in which they have added all 6 rare tRNA coding gene, and that strain is called Rosetta strain. So no need to check whether your gene is of AT reach or GC reach. You can use this. So they have added all means 6 tRNA coding gene in that strain. So Rosetta strain is perfect strain to overcome the codon biasness, effect of codon biasness. This is a self-mutant strain. So scientists, as you know, generally when we produce globular protein or membrane protein in E. coli, so E. coli doesn't survive. So this group of scientists, they were trying to produce globular protein in E. coli, membrane protein in E. coli. So E. coli, as usual, died, because E. coli, due to that toxicity, E. coli doesn't allow that production. So plasmid will be expelled out. So cell will die if you grow it in presence of antibiotic, because plasmid is not existing. So plasmid has this antibiotic resistant gene. So due to absence of antibiotic resistant gene, that transformed cell will not grow. So usually they tried to perform that experiment. So after a few days, they observed few colonies in the plate. Means that colony, that E. coli, survived the toxicity of membrane protein or globular protein. Means that strain is resistant strain, self-mutant strain. So when they sequence their genome, they observe that there is mutation in the promoter, and that causes ultimately this survival of the transformed cell, E. coli cell, in which your recombinant vector is present, and that recombinant vector globular protein coding genes are present. Now, so this is a mutation. So based on that information, C43, BL21 C43 information, this you can say NEB company have designed this strain, and so they have not added this T7 promoter, strong promoter, but they have added this tetraval ramonose promoter in place of T7 polymerase cloned under tetraval ramonose promoter, not that. Can be used for expression of toxic and globular proteins. So main purpose is in IPTG whether it will go express or it will not express. But in case of this ramonose promoter, expression can be increased or decreased, means it can be titrated by changing the concentration of the this ramonose, that is also a type of carbohydrate, you can say. So this is called LEMO21. So you can produce your membrane protein or globular protein in case of LEMO21. Arctic Express E. coli. So sometimes people try to reduce the culture temperature of E. coli in which you have added recombinant vector. Purpose is, or you can say motto of that reducing temperature is to decrease the production of your recombinant protein, so that chaperone protein present in that E. coli will take care of folding of your protein also, so that your protein will not form inclusion body. But sometimes people have observed that when you reduce the temperature of E. coli, so their chaperone activity also get reduced. So scientists have cloned they found that this you can say chaperone, which is, you can say, cold adaptive chaperone. So it will not reduce even at low temperature. This chaperone CPN10 and CPN60 will work efficiently even at low temperature. So that genetically engineered strain is called Arctic Express E. coli strain. Means scientists have added cold adapted chaperone coding gene in that E. coli strain, so that when you grow this E. coli strain at low temperature, this chaperone will take care of folding of your protein, so that protein will be insoluble for not in the inclusion body. BL21 star strain, there are multiple strains. Now I am coming to the disulfide bond formation. So as you know, most of the protein, they require disulfide bond formation for their proper folding, as you can see in case of insulin, in other proteins also. So we cannot, in normally E. coli cytoplasm will not allow disulfide bond formation in cytoplasm. So we have two options: either we transport our protein into periplasm where there is no reducing environment, so that your protein will get disulfide bond formation. But periplasmic space is very less, so your protein production will be very less. So scientists observed that, and they found that there are two enzymes, this thyroidoxine reductase and glutathione reductase enzymes are responsible for creating reducing cytoplasmic environment of E. coli. So scientists have mutated these two genes, and that strain is called origami strain. So now if you are using origami strain for production of protein which requires disulfide bond formation, you can produce. Their cytoplasm will allow disulfide bond formation, provided your protein must have cysteine. So it was going on that origami strain is good for production. Sometimes people observe that when there are multiple possibilities of disulfide bond formation, then their proteins are not in their activities. Recombinant protein produced in origami strain, their activities are not matching with the amount of the protein, means activity is less. So they observe that when there are multiple possibilities of disulfide bond formation, and we have allowed that disulfide bond formation in cytoplasm, so they form random disulfide bond formation, not proper disulfide bond formation. That's why due to random disulfide bond formation, all pairings are not like a native protein, so all the molecules are not properly folded. So to solve this problem, scientists have added in this origami strain, in which two genes are already mutated, they have added this disulfide bond isomerase gene. So that gene is producing the protein, and that is a type of chaperone which will take care of proper folding of the protein, means proper disulfide bond formation of the protein. So that strain is called sulfol strain, and that is very good strain if you are producing the protein with which require disulfide bond formation. So there are multiple combinations of this you can say origami strain and this sulfol strain. There is no strains available still to produce glycosylated protein in E. coli, but some groups are trying to produce N-glycosylated protein. So they have produced some genetically engineered E. coli strain, but that strain is as per my information, not commercially available. So still we are depending upon eukaryotic system for production of protein if that protein requires glycosylation. So first strategy we have discussed. This is summary of this strain. We will discuss next strategy by changing the vector in next video. So keep watching and keep in touch. So these are diagrammatic representation of that E. coli strain which we are using for different during different problems, you can say, when we face the problem. So thank you.